My name is Eike. I will be the chair of today's session, and I'm a global product manager for the spatial biology solutions at Miltony, based in the headquarters in Germany. I will give you a short introduction into the Miltony spatial biology solutions first, so just 10 minutes from my side. And then I will hand over most of the time to our two speakers who joined us today. So Christian Seitz and Sophia Scheuermann are here, and they will present how they apply the MACSima in the preclinical setting. So Christian is the head of stem cell transplantation and cell and gene therapy program at the KITS in Heidelberg, and Sophia is a postdoctoral researcher and the lab head in the Seitz lab, and she's also the PerVision scientific coordinator at the University Children's Hospital in Tübingen. Okay, but first, a short introduction into the spatial biology portfolio of Miltony from my side. So historically, Miltony was founded in 1989 by our founder, Stefan Miltony, and by now we have more than 5,000 employees, so quite a big biotech in Germany, and we have actually quite a lot of solutions for everything that you basically want to do in the lab. And you can see here on the slides, we have some sample preparation solutions, some solutions for cell isolation, cell culture, spatial biology solutions, all the way up to our highly automated cell processing platform, the Prodigy, for CAR T cell manufacturing. If you are interested in any of the solutions, since we are focusing on the spatial biology part today in the Spotlight Theater, if you have interest in any of the other solutions, please visit us at the booth number 4627. We have all the solutions present there, and for each of the solutions, we have experts there that can answer your questions and are happy to discuss with you. So today, as I said, I will focus on the spatial biology portfolio, and we have, on the one hand, our ultra microscope platform, so our 3D imaging platform that is a light sheet technology which lets you image large specimen in 3D. And on the other hand, we have our MACSima platform, our multiplexed imaging technology, which uses cyclic immunofluorescence to provide you with spatial data of thin tissue sections. So I will focus on the ultra microscope first. As I said, it's a 3D microscope using light sheet technologies to image cleared samples. And it can image basically everything that you throw at it, from small specimens, let's say organelles, but also up to large specimens, entire cleared mice, cleared rats, or even a human kidney. So the sky is the limit for you. Whatever you want to do, you can throw it at the ultra microscope platform. You see here an example of a data set that was acquired with the ultra microscope platform. You can see that you get a nice 3D reconstruction of your entire specimen to not miss anything in your sample. Now, the ultra microscope is actually the fastest light speed technology on the market, the fastest light sheet technology on the market. And our customers actually are using it for quite some years now. It was the first light sheet microscope that was commercially available. It has undergone quite some iterations now. It's now very automated, very fast. And over the years, our customers applied it to a plethora of different applications, which some of them you can see here. And also, since the data that you can produce with the ultra microscope is quite stunning, it also made it to the front pages of quite some reputable journals so far. And I'm very confident that it will continue to do so. Now, in pure Miltony fashion, we do not just focus on providing you the instruments or the reagents for an application, but we always have the whole workflow in mind. So we, of course, provide you with the ultramicroscope instrument, the Blaze or the Coros system, depending on how large the samples are that you would like to image. But we will also provide you with the necessary reagents to prepare your sample, so our clearing kits to clear the sample and apply the light sheet technology. We have a portfolio of 3D immunofluorescence antibodies to stain your sample. And after you acquired your data, we also have our MACSiQ View 3D large volume analysis software that lets you process the data. So we cover the entire workflow with our platform. And last but not least, we are also quite heavily using our own technologies at Miltenyi. And we have quite a few experts in-house, and they are more than willing to support you with your application. So we also offer scientific and technical support for all the platforms that we provide. Now, the second pillar of our spatial biology portfolio is actually the MACSima. The MACSima is a multiplexing platform that uses cyclic immunofluorescence of both RNA and protein detection. And basically for antibodies and protein detection, the sky's the limit. Yeah. Okay. Yes. So basically there's no limit how many antibodies you can stain on your tissue sections. It's up to you what you want to apply. 200 plus is mentioned here. We tested a lot more than that in-house. Now, a quick word on how this is actually applied on the MACSima. So we have our mix technology, so MACSima Imaging Cycling Staining. And I will just briefly explain what this means. It's actually a quite easy process. So you have 3 steps. You have, first of all, the staining of the sample with your antibodies, 3 at a time for the MACSima, directly conjugated to fluorophores. The system takes an image of your stained sample, and once this image has been taken, the signal from the antibodies will be erased, either by photobleaching or by an enzymatic process for specific antibodies that we have in our portfolio to get rid either of the fluorophore or the entire antibody if you are worried that maybe your epitopes are blocked for the subsequent cycles. Once this has been done, you can go to step 1 again, stain the sample again with another set of antibodies for different targets, and cycle through this as many times as you want. And all of this is fully automated on the MACSima, so there's no hands-on required for this entire process. And once you did this with the MACSima, you of course get these images for each of the markers that you chose in your panel. And after the experiment is over, all of the markers were stained, imaged, and put together in a data set. You can put all of the markers together and get the full insight into your tissue sections for all of the different markers that you chose and really deeply phenotype what is happening in the tissue. Yes, very important to note is also that the MACSima cannot just stain antibodies and detect proteins, but it is a spatial multiomics solution. So with our RNA guide technology, we can actually detect RNA and protein on the same section. And you can see an example here on the slide. So we stain RNA and protein on the same section. The RNA detection also follows our mix principle, which is very gentle to the tissue, and it preserves the tissue, so you get full access to the tissue also after the experiment was run. And you can see you can apply then, of course, the tissue to downstream assays. And here's an example where we did an H&E staining after the RNA and protein detection was done on the MACSima. So the tissue is very well preserved. Similar to the ultramicroscope, the MACSima is also a platform solution. So not just the instrument is provided to you. We also have MACSima sample carriers, so different sample carriers with different sizes, dependent on how large your samples are that you actually want to image. We have a catalog of antibodies that are actually pre-tested for the MACSima application. So in a customized manner, you can decide on antibody per antibody base what you would like to stain and put together your panel yourself, or we offer also predefined antibody panels to make it easier for you. For the RNA, we also offer an RNA predefined panel, but mostly it's a design product. So you can choose freely which target you would like to stain. You just give us the target, we design the probes, and then off you go, and you can detect whatever you want to detect on your tissue. To round it up, we also have our MACSiQ View analysis software, which is a very sophisticated and easy to use analysis software specifically tailored to analyze spatial biology data. So you can import just the data from the MACSima and then can do cell segmentation, you can do gating, you can do hierarchical clustering, UMAP analysis, and so on and so forth. So it really gives you an encompassing toolkit to analyze your spatial biology data. And similar to the ultramicroscope, of course, we are also offering scientific and technical support also for the MACSima platform. Now, to maximize your data, we thought about the workflow. How can we put these two technologies together, the ultramicroscope and the MACSima? So our R&D colleagues actually came up with a workflow where you first use the entire specimen. Let's say you have a tumor, you clear the tumor, put this onto the ultramicroscope, you image the entire tumor, identify the section that you're most interested in the tumor, you get the section, put this on the MACSima, do the high-plex imaging, and later put this data into the 3D context that you obtained with the ultramicroscope to put the high-plex information in the most context possible. And if you're interested in how this works, how the data can look like, actually there's a QR code on the slide. If you scan that, you will be directed to our STAR method paper that explains in detail how this works. Otherwise, just search for the 3D, 2D workflow of Miltony and you will definitely find it as well. Now, these are the solutions that we offer so far. So all of what you saw, you can get from us and we are willing to help you and support you. But I would also like to share briefly 2 applications that we are currently working on to advance the spatial biology field. And you see here on the slide perfectly that we have expertise in both the multiplex imaging and also the 3D imaging. We have experts in-house that know exactly how to develop these workflows. So I'm pleased to say that to advance the spatial biology field, we are also working on a 3D spatial biology workflow that lets you really dive deeper in the tissue microenvironment. So you are not able to just use these thin sections that you currently use for the spatial biology solutions, and you would get such an image that you would see here. But we are also working on using thicker sections and then using really advanced microscopy to really scan through these thicker sections, applying, of course, our mixed technology for high multiplexing, and then later reconstruct the data to get really the most information out of your sample and put the information of entire cells and not cut cells into the 3D context. And we believe that this will greatly advance the spatial biology field once we come out with it. It's under development currently, but you should really stay tuned. So it will be an exciting journey. There's a second application that I would like to briefly mention, but will then later on be explained in more detail by our second speaker and also by my colleague Hans Whaley in how you can apply it and how the technology actually works. And this application is actually a spatial in situ sequencing approach that lets you use tissue and then really sequence which sequences are present in individual cells within this tissue, in which cells these sequences are and how they interact or how these cells interact with the tissue microenvironment. So as I said, this will be explained in more detail by Sophia and Hans Whaley, and I will stop here and hand over now to our first speaker, Christian, and Christian will tell you more about how they apply the MACSima in the preclinical setting.
Okay, good afternoon. It's my great pleasure to talk about some projects that we're working on implementing spatial biology into precision oncology workflows. I'm a pediatric oncologist by training, and we're very interested in implementing immunotherapies in the clinical context for multiple reasons. Just to give you some background, this is a slide that pediatric oncologists like to show. It's the survival improvement of different pediatric diseases, cancer diseases over the past, I would say, century. it's very impressive, but the truth is also that over the past 30 years, we basically did not improve outcomes. And it's particularly interesting if you look at patients that relapse or have refractory disease. We have really dismal prognosis for these patients, and we cannot really offer good therapy. So implementation of immunotherapy should hopefully help to improve survival rates. At the same time, our therapies highly depend on chemotherapy, radiation, very toxic therapies, and that come with a high degree of acute and chronic toxicities and morbidities. So we really hope that immunotherapy can reduce this burden from our patients. And we all got really hyped. Yeah, it's quite a long time ago already, when Emily Whitehead was treated as a first patient with her first child with CD19 targeted CAR T cells. So she received a single dose of these genetically engineered T cells and went in deep remission and stayed in deep remission. So what we learned back then is that a single infusion of engineered T cells can cause cure of cancer. And we made more as a field, made more progress over the past years. There was really promising and exciting data shared by our colleagues in Rome, demonstrating that GD2 targeted CAR T cells can be functional and can induce remission and complete responses in pediatric patients suffering from solid tumors. It comes with a little caveat that the therapy mainly works in low burden disease, but it's a really promising sign to move forward. I would like to draw your attention on a poster that we're presenting this afternoon, where we treated a pediatric patient with massive bulky disease with brain-specific T cells. So a really nice response, and you will learn more about that response in the second talk by Sophia. On the other hand, other immune therapies like checkpoint blockade that works really well in adult cancer did really fail massively in pediatric cancer. So we didn't see response with immune checkpoint blockade. And so the question that came up over the past years is, what is different about pediatric cancers and adult cancer? What can we learn to optimize our therapeutic strategies? And first of all, you can see that pediatric cancers are totally different cancer types, right? In adults, we basically see carcinomas, whereas in pediatrics, we see leukemias, brain cancers, and some kind of sarcomas and embryonal tumors. So they really differ from the tumor entities. And of course, they also differ from targets. So we need to understand which targets are present in the pediatric cancers. And they're also different from a point of microenvironment. So you all know that cancers can be grouped in different tumor microenvironments. And in the pediatric setting, we're more on the cold edge of this. So again, we need to understand better how these tumors are made up. We need to understand better tumor immunology, spatial composition of the tumors, and also the target landscape to develop and improve immunotherapies in cancer. And to that end, our group focused a lot on profiling and identifying and understanding pediatric cancer. So there are multiple technologies at hand that we could use in that context. So there are the old-fashioned stuff like immunohistochemistry, basically looking at individual markers, but it really doesn't allow you to look at the whole tumor comprehensively. The same is also true for flow cytometry. You can add more markers, but you lose the spatial context. We did a lot of single cell sequencing. Again, you can dive very deep into individual cells, but you lose the spatial context, and you also don't have the protein that is important as the target structure for immunotherapy. So in the end, we ended up with focusing a lot on multiplex imaging. We were very fortunate that we had a long ongoing collaboration with Milton Biotech and were able to beta test long time ago already the MACSima and develop different workflows on that machine. So Sophia, who's sitting here and presenting soon, has really established the technology and the whole workflow in our group. And you can scan the barcode that brings you to a paper that we published 2 years ago that really describes the development of this workflow. Just very brief, Sophia established a panel of 120 plus markers that really allows you in fresh frozen tissue to deeply profile all the different cells that are there, sub-phenotypes of cells, and also the therapeutic markers and markers that associate with different function of different cells. And then you can also just very briefly mention here, there's a really nice analytic pipeline that comes with the MACSima device. A software MACSIQ that really allows you to do these basic steps of analyzing your data. So basically, image processing, QC, automated segmentation, which is like the holy grail of spatial biology. And then you can use marker-based ways or unsupervised ways to annotate your cells and do basic single-cell analysis like neighborhood analysis and distance analysis. But you can also use the data set, the CSV file that is generated, and fuel it into the bioinformatic pipelines that you're used to work with and that you like to work with. So just to give you a little vignette on how these workflows work, this is a study that we performed over the past years looking at NAT midline carcinoma. It's a very rare and very devastating disease. If you look at the outcomes on the right, it's driven by a NAT fusion, so kind of simple genetic background with a very detrimental outcome. So we got hands on a cohort of samples, and we tried to establish a multiomics workflow to really deeply understand this very rare and precious sample. So we did, first of all, whole genome and RNA sequencing, and then used these samples, generated tissue microarrays, and used them for MACSima high content imaging. And we also did sections where we validated the staining of particularly markers of particular interest on classic IHC. And then, of course, we fueled it into the downstream analytic pipeline. To that end, Sophia established yet another panel because these were FFPE samples. So we came up with a panel of total of 82 markers that allow cell phenotyping, that included therapeutic targets and also allow functional subphenotyping of the cells. And maybe interesting for you, like the major core of that panel is available now as the RIA Screen IO plate. You can buy that. So just very briefly, an overview. So these are all the different markers. And then if you want to dive into a sample just to illustrate to you how the data looks like. So this is a classical HNA staining of the same slide. And then you can go in. This is a IHC for NAT. This is the fusion that is really defining the disease. Now we dive into the mixed data. So this is DAPI nuclei. This is very beautiful. I always love that extracellular matrix. So you can look at collagens and vascular structure to really understand the architecture of the tumor. You can identify the tumor cells. You can identify immune cells, subphenotype, and different kinds of lymphocytes, myeloid cells, and then basically build up this whole microenvironment within this tumor. And then you can, of course, segment the cells and annotate them. So this is an annotated map of a tumor, basically phenotyping every single cell in the spatial context. So then what we then first did is that we kind of looked for different cell type abundance. As you can see, with such a small cohort of patients, and that's basically what's there because it's a very rare disease, it is hard to discriminate just by looking at abundance of cells, which are the ones that do better, that do worse. So we teamed up with Javad and Dennis Shapiro's group in Heidelberg to really dive deeply into that data set to understand what's happening. And I'm just going to give you some highlights that I would say. So basically what Dennis' group developed is a workflow, or a tool for conditional neighborhood preference analysis. So basically looking from my understanding as a physician, so looking at basically what cells like to sit with what cells and how do they interact. So basically developing a score of interaction between different cell types. And what you can see in the middle is that basically MDSCs like to sit next to a tumor. They like to interact with the tumor, while CD4 positive T cells are kind of far away. And we see this negative correlation between tumors that have MDSCs interacting with the tumor cells and tumors where T cells got closer to the tumor. And what is interesting now, if you look just at abundance of cells, just looking at cells that have a lot of MDSCs, a lot of T cells, versus those that don't have it, you don't really see a survival benefit or difference between the cohorts. But then, if you go close and look at these interaction scores, you can really find novel ways to understand these tumors and to understand prognostic biomarkers in these cancers, even if you have these very small cohorts of patients. So I think this is really lifting this kind of analysis to a whole different level, from just looking what's there to really looking what is spatially interacting with each other. We also looked at target antigens because we thought it's a good chance to just add a couple of targets on the MACSima panel. So this is RNA-seq data. So we first, did RNA screening on RNA level to see what targets are there, and then really wanted to understand if they're also present on protein level on the cell surface and what's the heterogeneity in the target expression. So we looked at the data and could basically confirm that there are multiple interesting immune targets on these tumors that could be relevant for immunotherapy. Now focusing on one of our favorites, MB7H3 or CD276. So we can basically show that it's uniformly expressed on basically all of the midline carcinomas. And then Sophia went back to the lab, we generated CAR T cells against the target and did both in vitro and in vivo study, really, I think, very nicely demonstrating how powerful these cells are. If you look at the right, or is it the right? No, here it's the left, sorry. If you look at the left, so these are basically mice with established midline carcinoma tumors. And as you can see that the CARs that are specifically targeting this target that we demonstrated in the analysis are really potent to eradicate these tumors in these mice. Having that said, I would like to summarize the first part of the talk. So there's a good end-to-end workflow that we've established together with colleagues and partners at Milteney that allows you to work with basically validated antibody panels and workflow to analyze your tumors and really get most out of these precious tissues. And then in the second part, I would like to focus on something that me as a physician motivates or excites me even more is the question, can we implement this technology really in patient care? So what we did over the past years were a couple of little feasibility study. It's more anecdotally, but I'm going to show you how we're going to transfer this in more systematic approach. So the idea is, can we, when we get a tumor biopsy, use the technology to really learn from it and instruct therapies? And I would like to share a first patient that we did quite a while ago already. That was a 70-year-old female, osteosarcoma, lung metastasis, really bad situation. Actually was scheduled for tumor resection, but they couldn't resect the tumor, so they got a biopsy. We got it on Monday. We put it on the machine the same day. And basically 3 days after, we got a really nice data output. So just to show you, this is DAPI. This is the healthy lungs around the tumor biopsy, extravascular structure, and that's the tumor. Likely tumor cells. And then we look for different targets. I'm going to cut that a little short here. But we saw that, again, B7H3, but also GD2 was highly expressed on the tumor, uniformly expressed. There was not much immune cell infiltrate, which is probably associated with the tumor not expressing HLA class HLA class 1 at all. There was there was some myeloid infiltration around the tumor, but basically a cold tumor, but a clear target. So we went back to the clinic. We verified that with a GD2 targeted PET. So really demonstrating that GD2 is highly expressed on these tumors. And I have to say that we were way faster than the pathology report. And then we started the therapy and treatment, the patient with the treatment with the anti-GD2 antibody in combination with chemotherapy, and really saw a nice response, temporal response. Patient eventually deceased, but I think it's a good proof of concept or example that this kind of workflows can be implemented. Maybe very brief, a second patient, that midline carcinoma, 8 years old. And again, we did the same analysis. And what was interesting here, what we found is that this tumor was very T-cell excluded. We couldn't find any T-cells in the tumor. And unexpected, we saw high PD-L1 expression across all the tumor cells. And that patient was then treated with a combination of PD-1 blockade and oncolytic virus therapy to kind of heaten up the tumor. And again, we saw nice responses, again, only transiently, unfortunately. But I think another proof of concept that it's not just, looking at target, which is kind of boring, but also looking at immune context and spatial orientation could really instruct therapies. So to bring this to another level and to structurize that, we are currently working on implementing the technology in a very nicely established precision medicine workflow. So at the kids in Heidelberg, the Pediatric Oncology Center, we have the INFORM study, which is a Europe-wide precision oncology program. So it's basically like what all these precision oncology programs are. So we have got fresh tumor samples. We do whole genome, exome, metallome sequencing. We also have a drug sensitivity profiling along with that. But really the goal is to, get in, let's say, roughly 3 weeks to a target report and to really have a molecular target, molecular tumor board to advise rational therapies. So that is established and ongoing, and it's based on fresh frozen tumor tissue. So what we did over the past 2 years, or 1.5 maybe, is that we tried to implement this mixed imaging into this workflow. So the idea and the goal is to identify target antigens, but also to understand immunological signatures that might be targetable with immunotherapy. And also to support biomarker-driven clinical trials. And we ended up with a 62 marker panel. Now the cool thing is that we still have space. So if there's a new target around, we can just add this to this panel. So it's not a fixed panel. It's a panel where we can build on. And just very briefly, this is the targets and this is the panel. So we have a couple of markers that allow to phenotype tumor cells. And we have markers that allow to phenotype immune cells. We also look at HLA expression because I think this is very relevant in the context of TCR T cell therapies on the horizon. And we also look for immune-associated targets like immune checkpoints, but mainly on like direct targets. So it's basically based on what is there, what is soon to be there, what we think is interesting. We designed these panels to look at different targets in pediatric cancers. We are currently in a feasibility phase, so we have already screened 15 patient samples, and we now want to build up a feasibility cohort to see if we could actually, predict targets from this patient cohort. And just to give you an example, this is a 12-year-old patient with Paxlovid positive fusion-driven rhabdomyosarcoma. The patient was diagnosed at chemotherapy, had radiation, had relapse, had lung metastases, and then we got a sample from that lung biopsy. We did the standard inform workflow, not really identifying a suitable target in that patient. And then we used consecutive slides that were basically cut from the initial tumor when taking tissue for RNA and DNA isolation and run a mix run on that. And just to give you an idea how it looks like, so this is an actual patient sample from the immuno-oncology program. So this was DAPI. This is the lung. Again, it's a lung metastases, tumor cells. We look at T cells. We look at vasculature and macrophages, and then the sample was segmented and annotated. So we get a nice annotation of all the different cells in these samples and in the spatial context. And then we looked for targets, and this is kind of still like an old school pathology like scoring, but we're currently working on reference tissue to correlate target expression and to semi-quantify it. But you can see that there's a couple of targets expressed, B7H3, FGFR4, which was interesting to see because this is a common target in fusion-positive rhabdomyosarcoma. We also found PRAME expression. And it's nice that you see that PRAME is in the nucleus where it should be. And then we also looked at HLA expression, which was rather low, but also nicely, and then membranase staining. We can also quantify, so we can go for, semi-quantify like high, medium, low expression. And we can also look at different cells. For example, what we started working on is this demonstrating establishing scores for different immune cell infiltration, basically looking, for example, at tumor and invasive margins, so we can quantify T cells in distance to tumor cells or macrophages in distance to tumor cells. And what's interesting to see that we can also, put expression profiles on top of that so that we can learn which kind of cells are important for these tumors.
Yeah, hi everyone. You've already heard my name. So I'm Sophia, And thanks for the opportunity to give you some recent insights into how we think that next generation spatial immunotherapy monitoring by high-plex spatial protein imaging and in situ sequencing by synthesis could look like. So we're all part of the cancer field, and we all know that cancer is very often described and also treated as a disease of malignant cells. But actually, that's not the full story, because a tumor per se is not just an accumulation of transformed cells that are growing in isolation. Actually, they're complex dynamic ecosystems. And for example, they have extracellular matrix components, cancer-associated fibroblasts, and also, blood vessels forming in those tumors. On top of that, we also have immune cells that either attack or support tumor growth. And all of these components, they don't exist solitarily. They all interact with each other, and they're an ecosystem that you have to look together, like that you have to analyze together as a tumor immune microenvironment. Now, to make it even more complex, if you think about immunotherapies, for example, here TCR T cell therapies, you add another component to this whole system. And it really depends on where those cells end up in the end and if they're functional or not. And today we're going to talk about transgenic T-cell receptor T-cell therapy. So this is the main immunotherapy that I would like to pinpoint to today, to give you a quick summary of what this therapy is about. So you're taking the patient's blood, you isolate the T cells, and all of those T cells, of course, have an endogenous T cell receptor. Then you transduce a cancer-specific T cell receptor. So then in the end, those T cells, they express the endogenous and the transduced TCR on their surface. And then you multiply those T cells out of the body and reinfuse them to the patient, where they hopefully encounter cancer-specific peptides that are being presented on MHC molecules on cancer cells. And we would hope that when they encounter those peptides being presented on MHC molecules, that they act on the tumor cells, kill them, and modulate the tumor microenvironment. So what we do in every immunotherapy that we apply, it doesn't matter if it's antibody therapy, vaccines, or adoptive T-cell therapy, for example, we monitor those patients, right? So we do MRI and PET imaging, we collect systemic fluids like blood, urine, CSF, but we also sometimes get our hands on biopsies or resections that give us information about local modalities in those immunotherapy treated patients. What do we do then with those samples? So these are just some examples that we do in the lab to monitor all of the different specimens that we collect. We do functional assays, we perform flow cytometry, we look at histology, we look at gene expression and proteomics. We also do liquid biopsies. And why do we do this? Because in the end, we want to understand how those immunotherapies work. What are the immune cells that are there? In which state are those cells? Do we have clonal expansion? In general, how is the tumor immune microenvironment looking? And do we see changes that the tumor is forming during therapy? And in an optimal way, we want to do longitudinal sampling, so have multiple samples at different time points. And today I would like to show you why we think that multiplex tissue imaging can actually combine several of those modalities into one, and things that we studied individually before can be shown using multiplex tissue imaging. So I'm really happy that I can show you all of this on a patient example that Christian mentioned before, and that he's also going to present today in a late-breaking poster in 236 today. And I'm not a clinician, so I'm not going to focus on the clinical side that we were looking at in this patient, but I was interested in how can we monitor the therapy that we applied to this patient. So the case presents as following. There was a 17-year-old male that came into the clinic in Heidelberg last year. He presented with a relapsed refractory nephroblastoma with at least 5 lines of therapy that he went through. He had a progressive, bulky metastatic disease, and you can see on the left, there's this huge mass in the abdomen. You see the metastases in the liver, but there were also mets in the lung and in the CNS. So, as Christian already introduced you to the informed precision medicine pipeline, I don't have to do that again, but what we did and what we used this precision medicine pipeline for is check for targets that we can really use for therapy. And here's just one example where we looked at PRAME expression on RNA level of this patient. You can see the red dot is the expression level of this specific patient in comparison to all the patients that were enrolled in INFORM. So this patient had a rather high expression of PRAME, which we also confirmed on a protein level using immunohistochemistry. So having this in mind, this patient was then qualified to being enrolled or included in a named patient use in close collaboration with Emetics. So we used a TCR T cell therapy, and we produced those cells actually as a point of care manufacturing approach using the Clinimax Prodigy in Heidelberg. So you saw those images before on the previous slide. You see this bulky disease in the abdomen and the metastases in the liver. And 9 days after we applied those cells to the patient, you can see there's a diffuse change in the signal. It's a signal alteration and heterogeneity. You see some changes in the mets in the liver. And at that time point, we actually had the chance to get part of the abdominal, so peritoneal sample from this patient. This was first monitored using classical pathological assessment. So went through classical stainings and pathology, and you can see that we have a massive influx in CD45, so immune cells being present, and many of them are actually T cells. What we did next is we took the exact same sample and sectioned it for MACSima Imaging Cycling Staining for being able to look at the same patient sample using more than one protein on a slide. So we chose 44 markers that we stained to be able to characterize the whole tumor immune microenvironment. And just to give you a glimpse of how those, like the results of this patient looked. So the nuclei in DAPI, and then you see some extracellular matrix components. You see in light blue the vessels, and in dark blue, I hope you can see it, the lymphatic vessels. Then the tumor per se in white, and then we recapitulated what the pathologist also saw. We see many CD45 positive immune cells, which we then further categorized or subcategorized here in MDSCs and M2 macrophages, but we also checked for HLA class 2 expression on any APC that might be around in the tumor immune microenvironment. And we also checked for T cells. And we see the same picture that we saw before, but now we can also already discriminate between cytotoxic T cells in red and all other T cells in yellow. Monomer functional side, we already wanted to check on a protein level what are those T cells actually doing, and we looked for granzyme B and Ki67, so activation and proliferation. And to show this to you in more detail, on the left side you can see in purple that we have a high co-occurrence of granzyme B staining on those T cells, so they seem to be highly active. And on the right side, you can see the overlay of red T cells with green Ki67, leading to yellow double positive Ki67 highly proliferative T cells. What we did then next is what Christian already introduced you to. We did segmentation and cell type annotation, and we could now quantify all of the different cell types that we find for this patient. If we only look at the lower bar graph, we can see that it's mostly driven by T cells, like all the immune cells are mostly driven by T cells here. We also performed a simple distance analysis for this patient, and we're here now able to see that we can bin those different T cells in relation to their distance to the tumor cells, which just gives us a more detailed an informed way of looking how and where like our T cells resides in the tissue. So with this, I'm coming to the first summary. So we were able to analyze this sample of the patient that we treated with transgenic T cells using a 44 marker protein panel for mix. And we were able to get a huge high resolution of the tumor immune microenvironment composition. We segmented those images, did hierarchical cell type clustering, and identified 12 different cell types with a high prevalence of T cells. We already saw on the protein level that those T cells are highly proliferative and cytotoxic. We performed simple distance analyses to be able to characterize the neighborhoods of those T cells, but we have to bear in mind that at this point, we're not able to discriminate between endogenous and engineered T cells. So we teamed up with our wonderful collaborators at Milteney and applied the mixed technology for same-section spatial multiomics. So following this workflow here, we used our tissue sample, extracted RNA, performed bulk sequencing, and analyzed the data, and used the data for custom probe designed for RNA-Sky. And for the bulk sequencing, we used a new kit, which is the Milteney's TCR alpha-beta RNA profiling kit, which allowed us to really look for the different clones that might be present in this specific sample that we were analyzing. And we focused then on the most frequent clones of the alpha and beta chain that we found in our sequencing results, which in the end helped us to design an RNA-Sky panel or detection probes for the 10 most abundant CDR3 regions for alpha and beta chains. We also included a probe for the TA peptide sequence because we hoped that this would show us the engineered T cells that have those ribosomal skipping sequence. We added important T cell related chemokines and cytokines and also some key T cell lineage markers. And to come back to the workflow slide, we also then did a consecutive section of our tissue sample and analyzed this on the MACSima, first doing RNA and then protein detection, which now allowed us for having a deeper level of spatial analysis, right? Not only looking at the different cell types, but also at different, for example, gene expressions. And this is exactly what I would like to share with you. So we now can look at the gene expressions by cell type, and you can focus on the different cell types that I encircled. So we saw higher IL-6 and IL-18 levels, for example, on the macrophages and CAFs, and higher interferon gamma levels on our T cells. And to take this one step further, we were then analyzing the T cell clonotypes that we found in this specific patient. First, in the upper bar graph, you can see that we can pinpoint and now spatially find the T cell clones we had from bulk sequencing on a spatial level in all T cells. And we saw a distribution that showed an enrichment for TCR alpha 10, TCR alpha 3, and TCR beta 6. If we now confine this even more and only look at the clonotypes that we find within our 2A positive cells, so the engineered TCR T cells, we can see that we find this one dominant or the dominant clones of TCR alpha 10 and TCR beta 6 within our 488 2A positive T cells that we found. So if you look at this on a spatial level, you can now combine the information that you gathered about the different clonotypes. And you saw this image already before in Eike's talk. It's actually looking for the different clonotypes on a spatial level. And if you focus on the green T cells on the upper image, every orange dot would be the presence of a transcript of TCR alpha 10. The green dot would be TCR beta 6. And if they're together in one cell, they form a paired dominant clonotype. And actually, this is also highly significant. So we find a significant enrichment of TCR beta 6 and TCR alpha 10 in this patient sample. Last but not least, we wanted to see if we can find those engineered T cells in distance to tumor cells. And this is just like the first step. We're not saying that we detect all of our engineered T cells, but what you can see here is that the pink cells, which are the engineered T cells, seem to be further away compared to all T cells from our tumor cells. And if we pinpoint that to the yellow cells, which are the paired dominant clonotype engineered T cells, it shows the same trend. So to sum up the second part, we use the multiomic analysis workflow that allows for the detection of our engineered T cells and their spatial distribution in this sample from this patient. We saw clonal selection in the engineered T cells, enrichment that was significant for TCR alpha 10 and TCR beta 6. So I'm not saying that this is the end of the patient story, because this is what you saw before, right? Before treatment, huge masses of tumor. And it was quite a miracle to see that obviously those TCR T cells were actually doing something, because they were clearing up the whole tumor. As you can see on the lower image, on day 100 of therapy, after infusion of the cells, the metastases in the liver were gone and the tumor in the abdomen really shrank to a minimal residue. And to look at that, the metastases in the lung, you can see in the upper level, compared to the lower level, you would think that there's like only a partial response. So we still saw some nodules present in the lung, but we had the opportunity then to get a biopsy from those samples to see what is going on actually in those residual nodules. And if we first again do our monitoring via pathological assessment, the pathologist told us that there were no vital tumor cells left. And in the middle panel, you can see what pathologists describe as ghost cells, so no nucleated cells left. But we found the presence of still present immune cells in this sample. So now that we had those two samples available, we went back and discussed last year with the colleagues from Milteney how to make the most of those samples. And this is where the latest addition to the MACSima workflow comes into play. We analyzed those two samples using same section spatial multiomics using in situ sequencing. So the upper part of the workflow you already saw. We did bulk sequencing and designed our probes. But we then used the new workflow that is still under development for spatial in situ sequencing. And combined with the classical proteome analysis, we were able to really pinpoint and deeply phenotype our cells of interest. And just a quick reminder, you can ask about the details for like all of the Milteney colleagues, and also there was a poster on that. Sequencing by synthesis, how does this work? So in general, your probes bind to the RNA, and then when the probes are elongated and they come in, they have a fluorochrome attached to them, so the sequence is going to be extended, then imaged, so the fluorochrome is imaged, and last but not least, cleaved away. And we used exactly this. First, I want to show you this section that we image here. It's a whole slide image of our tumor sample that we did, compared on the top, the peritoneum, and the lung sample on the bottom. The protein signal, you can see here that we can recapitulate the different immune cells and the stromal compartments that we saw before. But now we're able to layer on top the sequencing results that we had with this new workflow adaptation. And you can see that we're again able to find our engineered T cells within those samples. And to be 100% sure that what you find here as a transcript can be pinpointed to a T cell, we, as I said before, included like key lineage markers for lymphoid cells and T cells in specific. And you can see that our 2A positive cells were actually highly enriched for those T cells markers. This just shows you that what we do here, the analysis, really is clearly a T cell, and we're not like looking at random distribution. And as a last feature of that, we were able to really functionally profile those engineered T cells. You can see the comparisons between the peritoneum, looking in yellow at the engineered T cells in the peritoneum, and all other TCR positive T cells, we saw a significant enrichment for IL-6 and granzyme B in those engineered T cells. And also if you compare the engineered T cells in peritoneum and in lung, we saw an enrichment for interferon gamma. And if you think back to the image I showed you at the very beginning, where we saw these high proliferative T cells, the one that are Ki67, we're now able to say that it's actually the engineered T cells that are higher proliferative than all the other T cells. And I think this is exactly giving you one idea of how combining protein and the RNA level of your transcripts really helps us to understand those therapies better. So coming to my summary slide, we were monitoring this patient using a multiomics spatially resolved pipeline for seeing our TCR engineered T cells using RNA-Sky detection probes in situ sequencing by synthesis and direct protein detection. We characterized the tumor immune microenvironment in super detailed way. We detected our engineered TCR T cells. We saw clonal selection and could spatially and functionally really dive deep into those engineered T cells. As an outlook, I mean, we generated lots of data and we spent quite some time now analyzing, but the story is not told until the end, right? So we want to track those clones over time for sure. And we want to look at not only what are those T cells doing in distance analyses compared to tumor cells, but also to all the other cells that we found, right? And with this, I'm already at the end, and I want to thank the whole lab and especially the doctors that treated our patients and all the wonderful collaborators from Miltony, the whole team here in Waltham, Emily, and everyone that was helping out.